I'm going to talk about then specific nutrient solution recipes, and then showing you some symptoms and talking about solutions to common nutrient disorders. And of course, as a reminder, you all know the reason that we're supplying these nutrient solutions are because there's essential elements that plants need, and they need them in different proportions. They're all important, they all have physiological activities, but we need to supply them in different proportions. And these nutrients either have to come from the fertilizer source or from the water source, because in hydroponics we can't rely on the substrate to deliver the nutrients as well. So I'm going to talk next about some different fertilizer recipes, and you'll see that most of these recipes you're using 2 or 3 different stock solutions, and this is because of the problem with incompatibility of fertilizers. So that means blending certain fertilizers can result in chemical combinations that cause a precipitate, or basically gunk, at the bottom of your stock tank. And I'll show a very detailed table of different incompatibilities, but one of the main ones that we want to keep in mind is calcium nitrate is not compatible with other fertilizer salts that have phosphates or sulfates in them. And when in doubt, do your own jar test. That is, take the concentration of nutrient that you would use in one stock tank and the concentration from another stock tank and combine them together and see if a precipitate does indeed form. So for example, the beakers here show a small jar test that I did combining calcium nitrate with a 5,6,12 hydroponic fertilizer that had like magnesium sulfate and potassium phosphate in it. And what you see is the jars on the right, those were at the stock tank type concentrations, so 1 to 100 times concentration, and indeed a precipitate did form, which shows us that we do need to use separate stock tanks and separate injectors to deliver those nutrients. And you can find very detailed tables, incompatibilities online. Here's one that I like to refer to. You'll have the link in your handouts. That's from the Ontario Ministry of Agriculture. So then this is why we often have either 2 or 3 stock tanks. So one of the stock tanks is going to have calcium nitrate, and the other stock tank will have our sulfates and phosphates. And sometimes folks will add 3 stock tanks if there's additional incompatibilities that they have to be careful of. So then what I'll do is I'll present 4 different recipes for nutrient solutions for leafy greens and for herbs. And then I typically think about using these in 2 different stages. So the first stage would be the seedling culture stage. This is where you're growing the seedlings in trays of Rockwool, Oasis, Jiffy, or whatever your growing substrate is. These would be you'd seed into the 1-inch cubes or 1.5-inch cubes. Prior to seeding, you always want to be sure to pre-soak the cubes first with tap water. Sometimes there can be salts that are in the initial seedling substrate, and you want to remove those salts. And then you want to pre-soak the cubes or that tray in fertilizer water so that you're always seeding into a substrate that has fertilizer. The temperature then is often 68 to 72 degrees, perhaps a bit warmer than that for the initial 1 to 3 days of germination. Then these would be irrigated often on trays that either have drip irrigation, ebb and flood, or hand, so that you're periodically fertilizing them. Often maybe only once a day or so when the plants are young and first germinating, up to maybe 3 times a day, depending on your environmental conditions when you're getting close to the transplanting stage. We use a complete fertilizer solution, and often we use it at a slightly lower rate, say maybe 100 to 125 parts per million nitrogen, and then all the nutrients would be in balance to that, with an EC of say 1 to 1.5. And as Chris said, pH varies somewhat by crop, but often targeting say 5.4 to 5.8. Then these seedlings are ready to transplant when we reach the 3 to 4 true leaf stage, which under great environmental conditions, high light conditions, will be about 10 or 11 days, but can take as long as 3 weeks. And so this would be the seedlings that are getting ready to transplant, and you see they're irrigated with drip irrigation. And these are basal seedlings shortly after they were transplanted into an NFT system. This was some work that came about from the Peters Laboratory in collaboration with Smithers Oasis, and they looked at several different hydroponic crops and optimized nitrogen feed rates according to whether you were in that propagation stage or in that transplanting and production stage. So notice for all of the leafy greens and culinary herbs, which are in the top half of the table, and then we have heavy feeders and light feeders in terms of the leafy greens at the bottom, we're looking at a target of, say, 125 parts per million nitrogen for the propagation stage, and then bumping it up to about 150, 175, and then perhaps up to 200 for some of the heaviest feeders, such as cabbage and kale. And I think what Chris talked about in the first presentation is really important. It's that we talk a lot about target fertilizer concentrations or target EC, but what we find in practice is that if you have a large enough reservoir of these nutrients, so you've got a large enough reservoir for your NFT or a deep enough pond in the deep pond system, if you have that large enough reservoir and you have continual supply of those nutrients through recirculation in NFT, that the absolute concentration isn't so important. Plants are good at taking up nutrients across a broad concentration range, but you just need to make sure that the total quantity that they need is available to the plants in that reservoir and that you are continuously recirculating them. So then we will next proceed to talk about 4 of these different recipes. So I'm going to talk about simple recipes, so using 1, 2, or 3 bags of nutrients. And in these recipes, you're typically starting with a base feed that has macros and micros. So a lot of these would be so-called hydroponic formulas. And then typically there's not enough calcium or nitrogen supplied in those base feeds. So you're adding calcium nitrate as well. So that would go into a separate stock tank. Because of the fertilizer incompatibilities that calcium nitrate would have with the phosphates and sulfates in the base feed. And then last, it's important to test your tap water. Some tap waters will have enough magnesium for your crops, but otherwise we need to add magnesium sulfate or Epsom salts. So I'll walk you through those different recipes, and then I'll also show you a complex recipe where you can individually cater the nutrients using 11 different ingredients. So the 1 bag example is Jack's Hydro Herb feed. This is a 16-4-17. With this particular product, there is some calcium nitrate in there, and then the other nutrients that are often supplied as phosphates and sulfates aren't supplied as phosphates and sulfates. They're provided as nitrates as well. And so we're able to, in this case, to use just 1 bag, 1 stock tank to get a complete nutrient solution. And however, it is quite low in calcium and magnesium compared to some of the other recipes that I will show you. So, and I'll show you how these all stack up compared to each other in a moment here. But because it is somewhat low in calcium and magnesium, it would work well if your operation had relatively hard water to begin with, that is pretty high calcium and magnesium in your tap water. Otherwise, you may need to supplement those nutrients using that second stock tank, a second injector. So for this particular recipe, if we're supplying 150 parts per million nitrogen, which is kind of our target for most of the leafy greens and herbs, we need to use 355 grams per 100 gallons of water. So that would be 100 gallons of water total that the plant is getting. If you're using a stock tank with a 1:100 injector, then that means each gallon in the stock tank needs to have 355 grams of this fertilizer solution. So that was the 1 bag. Then a very common alternative is this 2 bag solution, and there's various hydroponic base feeds on the market. The example that I'll show you is Jack's Hydroponic 5-12-26. And so in this case, that nutrient solution has sulfates and phosphates in it, so we need to keep it in a separate stock tank than the calcium nitrate. So in this recipe, you can use equal parts of calcium nitrate and the Jack's Hydroponic 5-12-26. so 284 grams of each of them. And then I'll show you how that stacks up, but this recipe also supplies 150 parts per million nitrogen and a nice proportion of the other macronutrients and micronutrients. Then a relatively new fertilizer on the market is HORT Americas. I also wanted to point this out because HORT Americas is a sponsor of the webinar. I've started using this fertilizer in some of our trials at Cornell University as well. So again, the 9737 would be a base feed, and then you would add calcium nitrate and then magnesium sulfate to it as needed. So there's label directions if you search for the HORT Americas Hydroponic 9737. So in this case, I wanted to get a recipe that again delivered 150 parts per million nitrogen. So what I'm using is tank A has 280 grams of calcium nitrate, and then that will be in a separate stock tank. Then tank B, which has the hydroponic base feed, the 9737, at 150 grams per 100 gallons of water. And then it also has magnesium sulfate or Epsom salts to provide the additional magnesium that we need. So that's at 160 grams per 100 gallons. So those three recipes are relatively straightforward. Then I want to mention the lettuce recipe that we've used extensively in our controlled environment agriculture group at Cornell University. And this is a Sonneveld solution, the lettuce recipe from Sonneveld. And this recipe uses 11 different ingredients. And by having those 11 different ingredients, you can almost individually cater those different nutrients. And you might ask, would you go through all of this work of combining 11 different ingredients? A lot of it gets to the control, like Chris was talking about earlier. You might start with your standard fertilizer program, but over time, some nutrients may creep up and other nutrient concentrations may creep down as the plants took up the nutrients in slightly different proportions. So in this case, you could start with this recipe, then through periodic testing of either your hydroponic ponds or your nutrient solution reservoirs, then you can adjust nutrients so that you can still maintain a target concentration. So in this case, we have, again, the two tanks. So we have calcium nitrate in one tank and the other fertilizer components that are compatible with that. So the ammonium nitrate, potassium nitrate, and the chelated iron form. And then in the other tank, we have the additional macronutrients and micronutrients that we need that are in the phosphate and sulfate forms so that they don't precipitate out. So then the big question then is, how do they all stack up to each other? So in the recipes that I gave you, we all supplied 150 parts per million nitrogen, and you see that here. So we have the 16417, that was the 1 bag solution, the 51226 plus calcium nitrate, that was the 2 bag, the HORT Americas 9737 with calcium nitrate and magnesium sulfate, and then finally the 11 ingredient Sonneveld solution. So you see nitrogen is the same. Phosphorus varies from 12 to 39 parts per million. Potassium varied from 122 to 210 parts per million. Calcium, leafy greens tend to be lower users of calcium, but I'd recommend adding more calcium and magnesium than the 1 bag solution. And like I said, testing your water source will reveal if you have enough calcium and magnesium in your water source, that you can count on that for it. And then you'll see how the micronutrients break down as well. In terms of the micronutrients, they're all rather similar. So all of these solutions I've been able to use effectively for growing leafy greens in my research plants at Cornell University. So in the second half of my presentation, I want to talk about common nutritional disorders and correcting those disorders. And I want to start showing some photos we'll talk about 4 or 5 different common disorders. So this first one is nitrogen deficiency, and the symptoms of nitrogen deficiency are yellow lower leaves and reduced biomass from the plant. And so I had an undergraduate student, Tanya Merrill at Cornell, that did an excellent job looking at setting up hydroponic plants minus one of the the 12 essential elements that we were adding in our fertilizer program. So the container on the left was our nitrogen deficient plant, and the lettuce plant on the right was our control plant. And you do see that reduced biomass in the lower yellow leaves. Often this is just an indication as well of having too low of an EC or not delivering your nutrients quickly enough to your plants. And here's what nitrogen deficiency looked like for basil. In this case, the control plant is on the left, and the nitrogen deficient plant, of course, is on the right, so much sparser growth, and then, of course, those yellow lower leaves. Correcting nitrogen deficiency is, of course, quite easy. It's typically just indicative of low fertility. So you would need to increase your fertilizer strength, making sure that nutrient delivery is sufficient to your plants, and monitoring your EC and adjusting this either automatically using the equipment that's on the market now, or testing it by hand and manually adjusting EC daily by either increasing your fertilizer strength by adding more stock solution, or if you need to decrease your EC if it's climbed up too high, by just adding clear water. And then as Chris mentioned as well, periodic laboratory analysis of nutrient solutions is really the most important tool that you have to make adjustments, especially if you're new to hydroponic growing. This is going to really help you learn about your growing system. So I think the most common nutrient disorder that I see in lettuce is leaf tip burn. This is where the growing points and the young developing leaves have necrosis or tissue death at the edge of the leaves. And ultimately this is related to calcium deficiency, but it's almost never because there's not calcium in the fertilizer solution. Rather, it's because the growing environment made it difficult to get calcium up to that growing point. So with calcium, it's the main driving force for calcium uptake by the plant is active transpiration, and then calcium can't be moved around in the plant once it's been delivered to a specific part of the plant. And so what we find is that in a humid growing environment with poor airflow, there's not a good driving force for calcium delivery to that growing point. And then this problem is also exacerbated with head lettuce as it reaches maturity and the leaves start to unfold over each other and cover each other, that there's localized very high humidity in that growing point, and it's difficult for the growing point to get enough calcium. So the solutions to this problem would be, of course, verifying that you do have calcium in your nutrient solution, but then typically the solution is to control relative humidity. So for lettuce, we prefer relative humidity to be 50 to 70% in a greenhouse, as well as have good airflow. And research at Cornell has found that paddle fans, that is vertical airflow fans instead of horizontal airflow fans, so driving air to move over those growing tips, really helps to reduce incidence of leaf tip burn. The other thing that we do is we often talk about the target for light for lettuce is about 17 moles per meter squared per day. That's the daily light integral. And the reason we say that is because if you have higher light levels, especially as that crop is reaching maturity, you're pushing the plant too fast, and so then calcium uptake to that growing point and the newly developed leaves is not able to keep up with how fast the plant is growing, leading to tip burn. If you have poor airflow, like you don't have those vertical airflow fans, then your light target should actually be lower than 17 moles, maybe on the order of 12 to 14 moles of light. And then, of course, I just wanted to point out at the bottom that, of course, the lower light that you have, the longer your days to harvest is going to be. So if you had to reduce your daily light integral to, say, 10 moles because you were worried about not having adequate airflow and too high of relative humidity, it's going to take you about twice as long to produce a marketable crop as it would at that 17 mole target. A separate disorder in lettuce that's often talked about when people talk about tip burn is really a different disorder. It's called outer leaf edge burn. So in this case, it's not the growing point and new leaves that are affected, it's the outer leaves that are affected. And in this case, on the edges of the leaf, especially where the veins end in the leaf, you see again this necrosis, so death of tissue. And what's happening here is typically guttation. So some of the the xylem solution from the plant is exuding the veins where the veins end in the outer leaves. And you'll see this often on like grass as well. So, and this condition is promoted by high salts, as well as, in particular, fluctuating humidity conditions. So high night relative humidity is what really drives guttation. And then if you have that low humidity, then the exudates from the leaf are often reabsorbed in that same area. And then what happens is that salts will accumulate in that area of the leaf where the veins terminate, leading to death of cells. So again, this would be another disorder that's not really directly related to the nutrient solution, but more related to the environmental conditions that you're growing under. And you can learn more about the tip burn as well as the leaf edge burn in one of our e-GRO alerts. Then moving on to just a few more nutrient deficiencies I wanted to describe are iron deficiency. This is interveinal chlorosis of the upper leaves. So this lettuce has been affected by iron deficiency for a pretty long period of time, but typically the lower leaves are nice and green, and then it's the upper leaves that between the veins they show yellowing. And here's a nice example of that in hydroponic basil. So the lower leaves, notice, were fine, the one by the hand, but the younger leaves are showing this yellowing between the veins. And typically, iron deficiency is a problem with pH that's too high rather than a lack of iron in the nutrient solution. We often find that as pH goes up above about 6.5, depending on iron form, it's less soluble in water, and then we start to get that iron deficiency. So do then use nutrient solution analysis through a commercial lab to check your iron concentration as well, and make sure that you're targeting about 1 to 2 parts per million iron in your nutrient solution. And I just wanted to point out that most of the hydroponic fertilizers that we use do use a chelated iron form, such as EDTA, EDDHA, or DTPA, so that they are soluble across a wider range of pH. With iron sulfate, we find that solubility really goes down fast after about 6.5, and then you can see from this chart with EDTA, solubility is pretty good till about a pH of 7. With DTPA up to about 7.5, and then EDDHA is soluble across that broad pH range. Then magnesium deficiency, which looks quite similar to iron deficiency in that it's interveinal chlorosis, but in this case the lower leaves are what are affected. And so you see here that yellowing between the veins of the lower leaves. And another symptom that you will see is also that the leaf edges, and this again would be on the lower leaves, can show a marginal necrosis. Looks a little bit like potassium deficiency, but then as it advances, you will see more of that interveinal chlorosis developing within the leaf. And so then this is really severe magnesium deficiency that you would hopefully never see in your operations. In our case, we grew these plants for 3 weeks with that magnesium deficiency. And basil is also very affected by magnesium deficiency as well. In fact, we typically give it twice the magnesium concentration in the nutrient solution because we've seen magnesium deficiency so often in basil. So again, that yellowing between the veins of the lower leaves, and you see that on the plant on the left. The plant on the right has had magnesium deficiency in our experiments at Cornell for 3 weeks, and so besides just the lower leaves showing that interveinal chlorosis that has advanced farther up the plant as well. So correcting magnesium deficiency, magnesium is sort of the opposite of iron in that it's less available at a low pH. So again, you do want to check your pH and make sure that it's not getting down below about 5.5 or so, or 5.4 or so. And so in this case, though, magnesium deficiency is most often just because of a lack of magnesium in the nutrient solution. And as I talked about some of the fertilizer recipes, you don't add additional magnesium sulfate. They're counting on your tap water having magnesium, but if your tap water doesn't have enough magnesium, you need to add that with magnesium sulfate. So our target magnesium concentration for lettuce and leafy greens is 30 parts per million, and then as I mentioned with basil, we've had better luck using 60 parts per million as our target. And we do have a research update on e-GRO that came out in October on symptoms of common nutrient deficiencies in hydroponic lettuce, and I believe late next week we'll have a new one coming out with the symptoms in hydroponic basil, so do be on the lookout for that. And we talked about testing your nutrient solutions and your water source at commercial laboratories as well. Tissue testing is the best way to diagnose symptoms of nutrient disorders as they occur in the leaves, because some of the disorders can look alike. So it's important to work with the commercial testing lab to test nutrient solutions and then do tissue testing as needed as well. And with that, I'd like to thank you for your attention.